simple course. So don't leave. I advise you, mainly we are in the summer semester, don't leave them for the last moment to study the phytochemistry. This will not succeed. I'm telling you, frankly, from the first moment. I advise you to read each lecture when we take it. I put on the Teams all the references that you need. The book is there available. The course outlines is available. The lectures are registered, so you can go back to them any time. And keep in mind that the third part, which is very important, the explanation of the lecture. Not everything is written to you in the lecture, so you have to hear me well to understand well. You can ask me any question at any time, either on the Teams. I put my phone and my WhatsApp on the keynotes موجود عندكم وبالتالي you can go back to it anytime you wish. واضح clear? So the rest is your responsibility to study and to ask if there is something is not clear. It is totally different as a topic from what we studied before, because we started to study the natural products, the metabolites, which are produced from the plant according to the pathway from which they are derived. This is what we studied. If you don't know, I will take you back to the pharmacognosy, where we started to study the primary metabolites, which also covered in the biochemistry. Okay, and in Phytochemistry 1, we started to study the secondary metabolites, the first part of the secondary metabolites, and here we will continue to study the rest of the secondary metabolites. And the most important part of the secondary metabolites, I mean the key drugs, the lead drugs, remaining for Phytochemistry 2. So you will see that most of the drugs that you will deal with them all of the rest of your life and your practice will be covered in this course. So you have to give high attention for what we are discussing. Metabolites, those products which are produced from the plant, and to make it easy for the plant for study, usually we divided the metabolites which are produced from the plant into two types: primary metabolites and secondary metabolites. Primary metabolites and secondary metabolites. Primary metabolites, those metabolites which are essential for the survival of the cell. They are important for any organism, to survive, so to live, to grow. So they produce what we call the primary metabolites. In other words, to produce energy, to produce other building blocks for the biosynthesis of many other compounds, we need primary metabolites. No one of us can survive without fatty acids, without amino acids, without nucleic acids, without carbohydrate, and so forth. All of these are called primary metabolites. This means simply that all the organism must produce, biosynthesize these primary metabolites. Otherwise, the cell will die. That's all. End of the story. On the other hand, if I ask any of you, Yusuf, I want to ask you, Why you are
Yes.
drinking coffee?
To gain caffeine, which will energize my body, my organism, maybe.
Looking for what?
For
For
energy, of course.
energy. Came from what? Came from caffeine, no?
Yes, caffeine is
Yes, yes, came
the
from caffeine. So
compound.
without telling me, without asking, you usually drink coffee, tea, Coke, Pepsi, whatever you want. You are looking for one one metabolite came from the plant. And this metabolite, in the case of the coffee and the tea, is the caffeine. Now, the caffeine is classified according to us as secondary metabolites, not primary metabolites. If we go back to the plant and we ask the plant what the tea tree or the coffee tree benefit from the caffeine, from the secondary metabolites, I will tell you nothing. Wow, very interesting, very important. Is it true also for the plant which produce morphine, as the morphine is secondary metabolite? Yes. Is it true for the Atropa belladonna, which produce atropine? Yes. Is it true for the Taxus brevifolia, which produce taxol? Yes. Is it true for the Nicotiana tabacum, which produce nicotine? Yes, and so forth. So we know well that the plant per se doesn't benefit from what? From the secondary metabolite. On the contrary to the primary metabolites, which are essential for the survival of the cell. So the question is now, so why the plant produce these secondary metabolites, as the plant per se doesn't benefit from these secondary metabolites? Very important question. Practically, we don't know exactly why the plant produces these secondary metabolites, but we have some hypotheses in order to explain why the plant produces these secondary metabolites. Look, I use the word hypothesis because we are not sure. And one of the following four hypotheses could be true, or all of them, or two of the four, or three of them, and so forth. In order, we are trying to explain why the plant produce these secondary metabolites. They are not source of energy like the ATP in the primary metabolites. They are not utilized by the plant for building block and so forth. So why the plant produce these secondary metabolites? The science say that the plant produce, the first hypothesis, these secondary metabolites as a waste product. As what? As a waste product, because this plant has no excretion system like animals or like you. So whenever the plant has excess amount of any element, oxygen, nitrogen, carbon, sulfur, whatever, the plant, to get rid of excess amount of these elements, biosynthesize these secondary metabolites, and it will usually put it in the vacuoles of the plant to get rid of them. And we, as humans, we came to them to utilize them for other purposes, mainly as drug. So the first hypothesis, that these compounds are waste products. They are waste products. The second hypothesis, no, they are preserved compounds. And here we can give an example about the nitrogen. During pollination, the plant needs high amount of nitrogen. So if we measure the nitrogenous compounds, the secondary metabolites, I mean, manufactured from the plant as secondary metabolites containing nitrogen, before pollination and after pollination, we usually note that after pollination, the concentration of these compounds will become very low, will be declined, which means that the plant preserved the nitrogen in the form of secondary metabolites, and upon need, it destructs these secondary metabolites, reutilize the nitrogen for the pollination purposes. The second hypothesis, which could explain this, that these compounds, the plant produce the secondary metabolites as a defense mechanism to defend itself against enemies. You are enemy for the plant, the insect is enemy, the animals are enemies, whatever you want, all of these enemies. And this is explain the presence of good percentage of toxic plants around us, of toxic plants. Why these are toxic? Why the plant produce these toxic compounds? To protect themselves against the any enemies. The fourth one and the last one is a source of drugs. Yes. The answer is yes. The tea produce caffeine in order to make you, زي ما حكى يوسف, feel energetic. And the Papaver somniferum produce morphine in order to make you utilize the morphine as a narcotic drug, and the atropine as anticholinergic drug, and so forth. So practically, could be one of these hypotheses true, or all of them are true, or more than one is true. Whatever the reason is why the plant produces these secondary metabolites, but we know well now that for us, the main source of drugs from natural sources, from plants, belong to the secondary metabolites rather than the primary metabolites. Look, we use sugar, oils, proteins as a food rather than to be as what? As drugs. But on the opposite side, most of the drugs which are available in the market, and those drugs that we will see راح نشوفهم during our study to this course, they belong to the second group اللي هم as secondary metabolites, as secondary metabolites rather than to be as primary metabolites. Now the question is how the plant start the biosynthesis of these complex compounds. If I draw now the structure of the morphine, for example, or the Taxol, and you look at the structure of this compound, you will see that this compound is very complicated in its structure. And this is usually, as you know, and this is not the first time that you see some of these secondary metabolites. And if you look at it as such, the structure, you will note that it is complicated structure, huge structure. Now let us understand the structure rather than to memorize the structure. I mean by this, let us see how the plant can start the biosynthesis of any of these compounds starting from zero. How the plant, and this is called what? The biosynthesis. The biosynthesis. In order to do so, the plants need three main factors. How many factors? Three. Pathway, building block or construction mechanism between brackets and specific enzyme. Not or, but and. Again, not or, but and. Before starting, if you take two plants, for example, thyme and parsley. Thyme, parsley البقدونس. If you don't know what is the meaning of parsley. And you smell both of them, you will note that the thyme has smell, it containing secondary metabolites which impart this plant its characteristic smell. While if you go to the parsley, it has no smell. What is responsible for this? Who is responsible for this? This is the responsibility of the biosynthesis process. And one of the three major factors اللي حكينا عنهم اللي هم the pathway, the construction mechanism or the building blocks, and finally the specific enzyme responsible to make the first plant, the thyme, able to biosynthesize the secondary metabolites between brackets volatile oils that we will see next time, responsible for the aroma, while the parsley couldn't biosynthesize the same components because it has been lost one of these three major factors, mainly here in this case is the pathway. And you can take this as example to see all of the other plants. For the other plants, other example, thyme and mint, both of them containing what? Aroma. Both of them containing what? Volatile oil. This means that both of them have the biosynthetic pathway. And they have the same building block. So why the aroma, the smell of the two plants is different? Here we will go for the third factor, the specific enzyme, and so forth. And because of this, look around you, no two plants are similar. I mean in the sense not of the taste or the color and so on, in their content of the secondary metabolites. No two plants are the same. Even that they may containing same or secondary metabolite from the same group as we see. So this is what we mean by the biosynthesis, okay, of the biosynthesis. The biosynthesis does not occur in the plant, occur in your body, occur in animals, occur in microorganisms, in fungi, and so on to live and grow. So let us start with these three factors, اللي هم the pathway, as I told you before, and the construction mechanism, and finally the specific enzyme. In general, the pathways which the plant utilize for the biosynthesis of all the metabolites, here I mean either primary metabolites or secondary metabolites, lies within one of the coming four pathways. The first pathway is called acetate pathway. It is called acetate pathway. Acetate from acetic acid, okay? Acetate came from acetic acid. This is the structure of the acetic acid. How many carbons? Acetic acid. 2 ماشي؟ 2 carbons. هاي الاسيتيك اسيد. طبعا بنحكي على بايوسينثيسيس احنا هون، وبالتالي كو انزيم. ما بنحط او اتش مش هيك؟ وبالتالي 2 carbons. This is the first pathway. But the first pathway for the biosynthesis utilized from the plant is called the acetate pathway. This pathway, the acetate pathway, is responsible for the biosynthesis of primary metabolites. انتبه, be careful about what I'm telling you. So, the acetate pathway responsible for the production of acetate pathway. يعني يا دكتور fatty acids, amino acids, nucleic acid, proteins, lipids, all of them, they came from the same pathway, which is called the acetate pathway. The question now, can any plant have the ability not to produce primary metabolites? It is not anymore living cell. It is not anymore living cell. It will die. So yes, it is obligation. Let me ask you the question in another way. Is there any plant or any organism, including your body, living cell, doesn't containing the acetate pathway? No, absolutely no. Why? Because the acetate pathway responsible for the production of what? Of primary metabolites. And the primary metabolites are essential for the surviving of the cells. خلاص. Stop and start new paragraph. Clear? The second pathway is called shikimate pathway. Shikimate pathway. Starting from now, okay, the other pathway responsible for the production of secondary metabolites, not primary metabolites. The first one is called shikimate pathway from shikimic acid. Now we will see the structure of the shikimic acid, and the shikimic acid is usually, this is the shikimic acid, okay, responsible for the production of all ring and aromatic containing compounds. This means that if you go back to what you study in Phytochemistry 1, you study the anthraquinones, they are what? Aromatic compound. You study the flavonoids, the phenolic acid, all of them, whenever we see, this is the general structure of flavonoids, as you remember, clearly, ring, aromatic compound, it is derived from where? From this pathway, which is called shikimate pathway. So the shikimate pathway responsible for the production only for secondary metabolites, and these secondary metabolites are aromatic secondary metabolites. Aromatic secondary metabolites. The third one is called mevalonic acid pathway. Mevalonic acid pathway from mevalonic acid or mevalonate. Okay, from mevalonate. So whenever we are talking about acetic acid pathway or acetate pathway, this is to make it easy for you. واضح clear بس مشان نعملها easy for to make you understand it. بدل ما نحكي كل مرة acetate coenzyme A نحكي acetate pathway shikimate instead of shikimate coenzyme CoA, but it is clear that we mean this. So the mevalonic acid pathway, which is 6 number of carbons, COOH, 1, 2, 3, 4, 5, 6, and here we have OH. This is the shikimic acid. This is the structure of the shikimic acid. The of the mevalonic acid. This is the structure of the mevalonic acid, And the mevalonic acid is responsible for the production of two major secondary metabolites. These secondary metabolites are terpenes and steroids. Terpenes and steroids. Look here, steroids. We produce steroids. The animal produce sex hormones, steroids, cholesterol, and so on. All of these are classified as steroid, and some plants produce the same of them, while only the plant produce terpenes. But we couldn't produce what? Terpenes. So in certain steps, the plant and the animal or other organism can produce the same compounds from the same pathway. In other words, your body, as we will see later on, in our body, we have the mevalonic acid pathway, which is responsible for the production of steroid and sex hormones. The last one of these pathway is the amino acid pathway. Amino acid pathway. We know well that the only source of the nitrogen is the amino acid, صح؟ وبالتالي the plant, any secondary metabolites which containing nitrogen, by default, it is obligatory that the starting material for its biosynthesis must be amino acids. And here we have very important group of secondary metabolites, maybe the most important group of secondary metabolite, the most important source of the drug, of the key or the lead drug, which usually containing nitrogen, and these are called the alkaloids. These are the alkaloids which came from this pathway. The acetate, we have covered it in the pharmacognosy and in the biochemistry. The shikimate, we have covered it in the Phyto 1. So now we will cover the last important two topics, the mevalonic acid pathway, terpenes and steroid, and the alkaloids. And the course will be divided equally between the first part, mevalonic acid, and after the mid, we will start with the amino acid derived compounds, which are the alkaloids. So I hope that now you understand. If we go back to the example of the thyme اللي هلا حكينا and the parsley, I'm telling you that the aroma, any plant which is aromatic plant, aromatic means containing volatile oil. If you smell it, okay, you will smell something like the coffee, like the jasmine, like the rose, like the mint, like the thyme, and so forth. All of them come from this pathway, mevalonic acid pathway, while the parsley has no smell. So easily you can conclude, and you tell me that, sorry, the parsley doesn't contain the mevalonic acid pathway, and because of this, it is unable to biosynthesize what? The volatile oils. Clear? واضح? But the first condition, the first thing, important for the any organism, mainly here we are talking about plant, to produce any secondary metabolite, the presence or absence of what? Of the pathway itself. The pathway itself. If the compound doesn't containing the shikimate pathway, it will not be able to produce anthraquinone or flavonoids or phenolic acid or any of the aromatic compounds. Clear? That's all. طب جانا أخذنا المنث and thyme. What you can conclude, Anise? Both of them, معي? Both of them containing what?
Aromatic.
mevalonic acid pathway. لأنه both of them, yes, aromatic, معناته برجع على pathway. They containing what? الpathway. طب why they are not similar? We will go to the second factor, which is what? The building block. usually as they containing the pathway, they containing the building block. Remaining what? The difference which make the plant smell, aroma differ one from the other, اللي هو the presence or absence of what? Of the specific enzyme.
Enzymes.
Yes, specific enzyme. And because of this, if you look around yourself, I will come back to the building block. مش إني يعني بس to make you understand the issue. No two plants around the earth are the same. No way. Why? Either building block, either pathway, or building block, or mainly the specific enzyme. The specific enzyme which is responsible for the accomplish of the biosynthetic process. Biosynthetic process. Okay. Now, so the very thing, I hope that you understand what what we are discussing. Again, four different pathways. These are acetate, responsible for primary metabolite. خلاص. نكتب من أول السطر. Secondary metabolites, responsibility of shikimate, mevalonate, and amino acid pathway. These are the four different pathways which are the plant utilize for the production of either primary metabolite or secondary metabolite. Here we have important question. Can the plant utilize more than one pathway for the production of one compound? Two pathways for the production of one compound? Yes. And this will take us to the second factor اللي احنا سميناه building block. What is the meaning of building block? Look at the wall in front of you. If you look at it, you will see it as one piece. Yes? But if you go back, when they started to build it up, it is blocks. Check 1, 2, 3, and 4. And the same usually happen in the plant, in which the plant make piece, other piece, third piece, and then it will connect them. These three pieces could came from one pathway or more than one pathway. Yes, the answer is yes. The plant, راح نشوف example now. We will see some examples at the end of this lecture. The plant utilizes more than one pathway for the production of one compound. In some cases, one pathway, in some cases, two pathways, and so forth, depending on the type of the plant. Let us collect all of this. Don't worry. Take it easy. Give me 2 minutes, and you will see that this complicated slide will become very simple and easy. So don't worry. Let us start. For the biosynthesis from zero. Suppose, imagine that I have a cell, a plant cell created now, just now. It will go for the photosynthesis. And you know that the photosynthesis is either light photosynthesis or dark photosynthesis, the using of chlorophyll and so on. So the first product which is produced as a result of the photosynthesis is what? Glucose. Check. This is, we know well that from the photosynthesis, the first product which is produced by this process is the glucose. Now, starting from this glucose, all the sugars, ketoses, aldoses, have 5, 6, 4, 3 carbon number. All of them will be derived from what? From the glucose. Dot. Clear? And obligatory, obligatory, it is an obligation, and you know this from the phytochemistry and physiology, that the glucose will enter the glycolysis process. This is occur in our body and occur in the plant. The plant enter the glycolysis process in the form of glucose 6-phosphate. Why 6-phosphate? Phosphate is also always a source of energy. So no biosynthetic process will be accomplished without the presence of what? Of energy. So wherever you see the phosphate, this means that it is a source of what? Of energy. In order to make the glucose 6-phosphate split into 2 units, the simplest sugar, we call them glyceraldehyde. Glyceraldehyde, okay, اللي هو the simplest sugar. 1, 2, 3. So 2 units of glyceraldehyde will be obtained from the glucose. Further oxidation will occur, okay, for this OH to give us اللي هو the glyceric acid instead of glyceraldehyde. And this glyceric acid will be the starting for the phosphoenol pyruvate production to give us to end up with the pyruvic acid. Still we are talking about 3 carbons. 1 2 3 مش هيك؟ 1 2 3 different carbon, and the pyruvic acid under a specific enzyme will lose 1 decarboxylation process, decarboxylase enzyme. This is the specific enzyme, decarboxylase meaning the loss of 1 carbon. Okay? Decarboxylase. Carboxylic group will be what? Lost. How many carbon here? 1. We started with 3 and end up, pyruvic acid end up with 2, and this is the first biosynthetic pathway, which I called it before acetate pathway. Acetate pathway. Now, the acetyl coenzyme, as I told you before, either we use acetate, acetic acid, or acetyl coenzyme. So whenever we use acetate pathway, we mean acetyl coenzyme A. Clear? But instead of telling this long sentence every time, we use acetate. But you have to understand that we mean that the acetyl coenzyme have 2 choices now. Either it will enter the Krebs cycle, and by entering the Krebs cycle, it is a source of energy to produce 38 ATP unit, and some amino acid will produce during this process. This is from one hand. From biosynthetic pathway, the acetate itself, which is 2 carbons, don't forget this, this is the acetate, 1 carbon, and this is the second carbon. This is the structure of acetic acid of the acetate. Okay, so the acetate now is considered the main pathway, in part the building block for the biosynthesis of many of the primary metabolites. To make it easier for you, go back for biochemistry and remember any of the fatty acids. Any fatty acids. Always, if you remember, that the fatty acids are even or odd number. مين بتذكر؟ even شيء C12, C14, C16 and so on. Why? لأنه الأسيتيت the starting group هلا راح تشوفه كمان مرة, this is 2 carbons, so it is difficult to be even number. So this is the first pathway, the acetate pathway. The acetate pathway from acetic acid. Will acetate come carbon? Again, not 2. This is the acetic acid. Here we write acetyl CoO and so on. This is other things. 1 carbon, 2 carbons. So the plant, whenever you need 2 carbons, it will go for the acetate. For the acetate, and now the acetate pathway responsible for the production of a huge number of what? Of the primary metabolites. Primary metabolites. The second pathway, the shikimate or the shikimic acid, this is the structure of the shikimic acid, per se came from the glycolysis process by a reaction between the phosphoenol pyruvate and erythrose 4-phosphate. This is sugar, came from the glucose per se, containing 4 carbons, 4 different carbons. Erythrose 4-phosphate will react with the phosphoenol pyruvate to give us the shikimic acid. Now, the shikimic acid is responsible for the production of all the aromatic compound, including the aromatic amino acid. Including what? The aromatic amino acid. You know that we have only 3 aromatic amino acids: phenylalanine, tyrosine, and the tryptophan. Tryptophan containing indole. These are the only fatty acid from the essential fatty acid which are aromatic. And as they are aromatic, without saying any word, they came from where? From the shikimate pathway. While look, the aliphatic amino acids, they came from which pathway? From the acetate. During the glycolysis process, okay, all of these amino acids, and when the acetate entering the Krebs cycle, it will produce all the aliphatic amino acid. If you count these amino acids, with these you will find them 20 carbons, the essential amino acids. So clearly now the acetate produce the aliphatic amino acid. The shikimate produce what? The aromatic amino acid. This from this part, amino acid. The acetate now will enter many reactions to produce many of the primary metabolites, while the shikimate is responsible for the production of aromatic secondary metabolites. The last pathway, or the other pathway, the third pathway is the mevalonic acid pathway. This is the structure of the mevalonic acid. Mevalonic acid per se, mevalonic acid نفسه هو, it came from 3 acetate units. كم واحد حكينا how many carbon الاسيتيت؟ هلا كنا نحكي 2 carbons.
2 carbons.
So, multiplied by 3, 6 carbons.
6.
مثل mevalonic acid, how many carbons? 6 carbons. Mevalonic acid responsible for the production of 2 big group of secondary metabolites. These are called terpenes and what? And steroids. It has nothing, as you see in front of you, with the amino acids. To produce no amino acids. خلاص, amino acid, they are products from the acetate and what? Shikimate. Mevalonic acid has nothing to do with what? With any of the amino acids. Neither aromatic nor aliphatic amino acid. These amino acids, either they are aliphatic amino acid or aromatic amino acids, the plant for the production of alkaloids, حكينا, because they're containing what? Nitrogen, the plant will utilize these amino acids as a 4th pathway for the production of these. Clear? As I promise you, this became easy. I don't ask you to memorize it. I want from you to understand, not to memorize this. So these are the 4 pathways. Let us now to end up with the building block. What is the simplest building block? C1, no?
C1.
The simplest is what? C1. The plant needs C1. Needs where? For example, if the plant has OH and we want to make it OCH3, this is how many C? 1. NH2, I want to make it NHCH3. This is what? C1. So the plant, please keep in mind the following, that what we are studying is the biosynthesis which is occur in the plant, not what we are writing on the desk to do. Yani this is occur in the plant as is. This is what the plant is doing, not what you want the plant to do. The plant will not hear you, no? Clear? In order to make you understand some examples, as I will give to them to you as after a while. Okay. So the plant want to biosynthesize how many? C1. Usually the plant utilize this amino acid methionine because it has a good leaving group here. Don't forget that the adenosine will attach to this sulfur here as coenzyme. عشان هيك بنسميه because of this usually we call this S-adenosine. Here the adenosine methionine. From where the methionine came? This is the methionine. Obligatory it came from the Krebs cycle. So the plant has it always from primary metabolite. So the plant will utilize the S-adenosine methionine. Okay, this is good leaving group, expulsion of this CH3, and the CH3 will go and to give the S1. And usually we use it SAM as abbreviation. SAM, SAM, S-adenosine, S, this is this, adenosine methionine. Okay, coenzyme, facilitator for the enzyme. Okay, if I ask you, the plant after C1 we have what? C2. ما بتعرفوش تعدوا. C1 after C1 C2. Directly, some of you will tell me 2C1, doctor. No. No. The plant will not go for 2-SAM to make C2 because the plant already has what? The acetate, the acetic acid. I told you many times that the acetate, how many carbons? 2 carbons. And it is always ready. So the plant, when the plant needs C2, it will go for what? Acetyl coenzyme. Okay? Acetyl decarboxylase, reduction here of this group. Rest is what? Is the C2. Now, if the plant needs C3, 1 acetate and 1 what? SAM. C4, 2 what? C2. C6, 3. C8, and so on, the plant. For this, look, this is how many carbon? C2. And this is C2. And this is C2, and so forth. Because of this, this is my answer to the question about the fatty acid. While the fatty acid always are what? Even number, and not what? Odd number. Go back. Lauric acid, C14. Stearic acid, C18. Palmitic acid, C16. Why they are not C15? C13, C7, we haven't. Why? Because the plant will take this unit, C2, C2, and it will put one with the other attachment, just this. واضح, clear? This is how the plant. Now, what about C5? شو رأيك? How the plant can make C5? 2C2 plus 1C1, no? Can this work? The answer, no. The answer, no. If you want to write this on paper, on the lab, okay. But what is happening on the plant? That the plant will not go for this 2C2 and 2C1 to make the C5. This is wrong. the plant will go for the mevalonic acid. And I explained the mevalonic acid for you, C6, which came from 3-acetyl. By one step decarboxylation, look, decarboxylation, it will give us C5, this unit, which is called isoprene. Which is called what? The isoprene unit. In other words, انتبه, to make you understand, the plant which containing acetate pathway unable to biosynthesize what? C5. And because of this, I told you at the when I start this lecture, what is the difference between the menthe and parsley? The menthe containing mevalonic acid pathway. The parsley containing acetate, but not what? Mevalonic acid. So it is impossible to biosynthesize what? This isoprene. I hope clear. Okay, let us go again. C6, C3. The amino acids are there. Okay, the amino acids are there. Just the plant make either decarboxylation. Be careful about the enzymes. انتبه للانزيم اللي عم بحكيها. Decarboxylation or deamination. If the plant wants C6C2N, it will make what? Decarboxylation. If the plant wants C6C2 or C3, reduction of this C3, C6C3, the plant will make what? Deamination. And these amino acids, phenylalanine and tyrosine, are available in the plant. No? From the shikimate pathway. So the plant, in order to biosynthesize this, it must containing what? Shikimate pathway. Indole, the same story as you see, either decarboxylation or deamination. And starting from the C6C3, the plant by reduction can make C6C2. By further reduction, it makes C6C1. Indole, decarboxylation. Suppose that the plant doesn't containing the specific enzyme decarboxylase. The answer is always the plant is unable to biosynthesize this. These are building block. This is called what? Building block. This is building block. This is building block. This is building block. Now the plant will take more than one of these building block. C4CN, it will go for ornithine amino acid. Decarboxylation, deamination, to give us the C4N. And then what? Cyclization. And then the cyclization. C4N. C5N, lysine containing one carbon additional. So it will give us the C4N. Clear? The question of saturation or unsaturation, the presence of double bond and absence of the double bond, came as a later step. To end up, these are some examples. Let us, for example, take any one of them. Let us take this. Okay, this came from where? Now the plant making the building block, and at the end of the day, this is C6. امشوا معي, follow me. C6, 1, 2, 3. C6, C3. Yes, one unit came from phenylalanine or tyrosine. Tyrosine, لأنه في OH here. Okay, amino acid tyrosine. And this is, okay, C2, C2, C2. So the plant utilize 1 unit of C6, C3 and the 3 unit of C2 in order to make what? This compound. In other words, the plant make this unit again alone and this unit alone, and then make a combination between the two. Look at this, for example. If you look at the structure, you will feel that it is complicated. No, it is easy, simple. What we call this unit? 1, 2, 3, 4, 5. Isoprene. Well, this is 1, 2, 3, 4, 5. Isoprene. And this is 1, 2, 3, 4, 5. Isoprene unit. What the plant made? This unit plus this unit. Look, without the addition of any carbon. Bridge, just bridging. And this unit with this unit. And this unit with this unit. The plant attach them together to produce this compound. Look here, it is the same. This is isoprene unit. 1, 2, 3, 4, 5. And this is what? 1, 2, 3 attached to them. But without the addition of carbon. This is always when you see this C2, 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 it came from where? From the acetate. The same story for what? For the C6, C2N. This is C6, C2. Decarboxylation, deamination. Here, just decarboxylation. Attach the nitrogen to the carbon and so forth. This is, I think, now you are easily isoprene. Well, this is indole C2N. Attach this unit to this unit. As you see, that the plant utilize either one pathway for the biosynthesis of the compound, like this example for mevalonate, or in other cases, it can use what? Here, shikimate and acetate. It can utilize more than one unit for the production of one compound, as you see. So the plant can utilize either one pathway or more than one pathway for the synthesis of one compound. I hope that it is clear, give you an idea about the biosynthesis, how the plant start to biosynthesize these complicated compound pieces, and each one of these pieces came from different pathway, needs different building block and enzymes, and it will attach it. When you look at the compound for the first time, you will see that, oh, it is complicated, but if you can understand it by this way, it becomes very easy, very simple, as you see. في أي سؤال؟
دكتور
Any question؟ تفضلي.
دكتور هلا بس بالنسبة للستراكشرز لازم نحفظهم ولا إنه نفهم؟
راح يجوكي 1 by 1. طبعا
نعم.
بتحفظيهم. راح يجوكي 1 by 1 يا لما.
لا قصدي مش البيلدينج بلوكس، قصدي ستراكشرز موجودين مثلا بالسلايدات أو هيك.
طبعا يس طبعا. راح يجوكي 1 by 1. All of these راح يجوكي. Just now I am telling you, I'm giving you example.
تمام اوكي.
اما later on yes.
اوكي.
كل الامتحان لما structure. من هلا بحكي لكم. الفايتو كيمستري والميديسينال كيمستري structures. هذا معروف يعني. anyway طب ما تحمليش حالك لحالك، انا بحفظك اياهم بهاي الطريقه السمبل السهله اللي يعني تصيري فريندلي يوزر. دونت وري.
دكتور سهل الحمد لله تمام.
keep them ان شاء الله. في اي سؤال حدا حاب يسال؟ اوكي بقدر اسالك. السلام. يس يوسف.
I wanted to ask when you told that we have deamination and decarboxylation, when we in our structure have nitrogen like in indole, we call it deamination happens too, yes or no, or just decarboxylation? If we have one nitrogen in structure, not NH2, of course.
The plant usually utilize the specific enzyme, okay, to remove either the carboxylic acid or the amino group. This is what you mean by reaction, which is called deamination, and the enzyme is called deaminase enzyme.
Yes, but my direct question, like when we have indole and we have in structure like C2N, like does it deamination happen or no? Because we don't have an H2 group, but we have nitrogen one. That's why deamination.
No, it is yes. Okay, I got your question. This is two-step reaction: deamination, reduction. Deamination,
yes. Thank you very
reduction.
much.
And then after the deamination, if the plant needs to remove the hydrogen, this is a reduction reaction. I didn't show you here all of the reaction because my interest is in a certain, because each one of these many steps, no? I don't mind of these steps. I don't want you to memorize them. I want you to take the major area. But for your question, yes. First of all, deamination, or decarboxylation and then reduction after this. In this case, it will remove the hydrogen from the compound or from the amino acid. أي سؤال؟ any other
Thank you
question?
very much, Dr.
Okay. Okay. نشوفكم بكرة إن شاء الله. باي باي. بس please again روح ادرس المحاضرة today not tomorrow. إن شاء الله. الله يعطيكم العافية.
يعطيك العافية دكتور.
Goodbye,
يعطيك العافية
Dr.
دكتور.
Goodbye.